Welcome again to BioBits and Bytes.
Great to be here.
Today, we're diving deep into something really fundamental happening inside your cells specifically, within those powerhouses known as mitochondria.
Yeah, the energy factories.
Exactly. And you shared some fascinating research on a protein complex called Prohibitin. Now, this sounds a bit like, I don't know, a security system or maybe even the scaffolding that keeps these tiny factories in shape.
That's a spot-on way to think about it, actually. If you picture cells as these bustling cities, right? Mitochondria are the power plants.
Okay.
And just like any power plant, they need structure, organization,
Yeah.
and prohibitin seems to be key for that.
So our mission in this deep dive is really to unpack what this prohibitin complex is, figure out where it lives inside the mitochondria, understand its jobs, and maybe reveal why knowing about it is, well, surprisingly relevant to how our cells work and stay healthy.
Exactly. We'll look at what it's made of, where it sits in that really complex folded inner membrane,
Right.
the many hats it wears, function-wise, and yeah, why it matters for our health.
And the research you mentioned uses some incredible techniques like cryo-electron microscopy, seeing these things frozen in time almost.
It's amazing stuff. Cryo-EM lets us see these molecular machines in their kind of natural state without too much distortion. It's revolutionizing how we see these structures.
And there's other research looking at its role in mitochondrial DNA maintenance.
Yep, the mitochondria's own little genome.
Cell survival, even disease connections, so a lot to unpack.
Definitely. These techniques are like giving us superpowered glasses to see inside the cell.
Okay, let's get started then. Let's explore the intriguing world of the human prohibitin complex. So first things first, what exactly are these prohibitins at the molecular level?
Okay, so prohibitins, specifically prohibitin 1 or PHB1 and prohibitin 2, PHB2, they're proteins found across a huge range of life. Bacteria, yeast, plants, us.
Wow, really conserved then?
Highly conserved, which tells you they're doing something, really fundamental. They belong to a bigger family, the SPFH family.
SPFH.
Yeah, it stands for stomatin, prohibitin, flotilin, HFLKC, bit of a mouthful. But the common thread seems to be that they all anchor themselves in cellular membranes somewhere.
So PHB1 and PHB2 are the mitochondrial members of this club.
Yeah.
And you said they team up. What's that partnership like?
Precisely. In the mitochondria, they don't go it alone. They assemble into this really big structure, a multimeric complex. It's huge, molecularly speaking, around 1 megadalton.
1 megadalton. Okay, that is big for a protein complex. What's the dynamic between PHB1 and PHB2? Are they like interchangeable parts?
Not really. They're very interdependent, like dance partners who need each other. If you lose one, the other one usually gets unstable and disappears too.
Okay. So they really need each other to form that big complex.
Exactly. They form the functional unit together.
And structurally, what are the key bits of these individual proteins?
Right, so they share some building blocks. PHB2 has what's called a transmembrane domain. That's the bit that anchors it into the membrane, the lipid bilayer.
Okay.
PHB1 doesn't have quite as clear-cut a transmembrane part, but it definitely associates with the membrane, probably by sticking close to PHB2.
Got it.
Both have a conserved bit called the PHB domain, that's the family resemblance part. And crucially, they both have a coil C terminal domain that's like molecular Velcro that lets them grab onto each other.
Coils. Okay, so that's how they link up. How do these pairs then form the bigger complex? What's the overall shape people first thought it had?
So they assemble into this ring-like structure, sitting in the inner mitochondrial membrane, the IMM, and poking out into the space between the inner and outer membrane, the inner membrane space.
Okay, a ring.
Yeah, early work, mainly with purified yeast proteins, suggested these rings were made of maybe 12 to 20 pairs, heterodimers of PHB1 and PHB2.
Right. A pair is 1 PHB1 and 1 PHB2.
Exactly. And these rings were estimated to be about 20 nanometers across.
20 nanometers. Incredibly small. How many of these rings are we talking about inside a single cell? Sounds like there could be loads.
The numbers are pretty staggering, actually. Estimates for a typical human cell, like a U2OS cell, suggest around 214,000 of these rings, assuming maybe 16 pairs per ring.
Wow. 200,000.
Yeah. And most of them, something like 75 to 84%, are found in the cristae membranes.
Those folds inside the mitochondria, right? Where the energy production happens.
That's the place. Increases the surface area enormously. So we're talking maybe 170,000 rings packed onto those cristae.
That's hard to picture. What does that mean for one single fold, one crista?
Well, estimates suggest maybe 11,500 cristae per cell. So if you do the math, it works out to about 15 prohibitin rings per crista, maybe 7 or 8 on each side of that membrane fold.
15 rings.
Yeah, and given their size, they only cover maybe 0.7% to 1.4% of the cristae surface area.
Such a small percentage, but clearly vital if there are so many.
Absolutely. It highlights their importance even if they don't take up much space.
So that early work gave us this ring idea from purified proteins, but seeing them in the mitochondria in their natural habitat, that must have been tougher.
Oh, hugely tougher. And that's where cryo-electron tomography, cryo-ET, has been such a game changer recently.
Right, the technique you mentioned earlier.
Exactly. It lets us get these 3D snapshots of structures inside the cell in their native environment, like looking through a window right into the mitochondria.
And what did they actually see using cryo-ET? Did it confirm the rings?
Well, they saw lots of distinct shapes embedded in the inner mitochondrial membrane, mostly in the cristae, as expected, but also sometimes in the smoother part, the inner boundary membrane.
Okay, distinct shapes. Not necessarily perfect rings then. What did they look like?
From the top, looking down, they did look roughly circular, about 20 nanometers across, so that matched the older ring estimates pretty well.
Oh, okay.
But from the side view, they looked more like convex shapes, like little bumps or maybe flattened domes, about 9 nanometers high, and they seemed to bulge out towards the inner membrane space.
Interesting. So it's sort of dome or bump, 20 nanometers wide, 9 high. How did they confirm these bumps were actually the Prohibitin complexes?
Good question. First, they checked other types of mammalian cells, rat brain cells, monkey kidney cells,
Mm-hmm.
and they found similar structures there too. That suggests that it wasn't just a weird thing in U2OS cells, but probably a conserved feature in mammals.
Okay, finding it elsewhere helps build the case, but how did they prove it was Prohibitin?
They used a really clever trick called RNA interference, or RNAi. Basically, you can use it to specifically reduce the amount of a particular protein in the cell.
Like turning down the volume on the Prohibitin gene.
Exactly. They turned down either PHB1 or PHB2. The idea was, if these bumps are prohibitin complexes, reducing the building blocks should mean fewer bumps. Right?
Makes sense. And did it work?
It did. When they lowered PHB1 or PHB2 levels, they saw significantly fewer of these convex structures. That was pretty strong evidence.
Okay, that's compelling. I think you mentioned they did one more thing with fluorescent tags.
Yes, exactly. They used versions of PHB1 or PHB2 that had a little fluorescent tag attached, like putting a tiny light bulb on them.
Right.
So in cells where they'd already reduced the normal prohibitin, they introduced these tag versions. And guess what? They saw extra little densities, like tiny extra bits, sitting right on top of those convex shapes.
Ah, and those extra bits matched the size of the tags.
Pretty much, yeah. It was like catching the tag protein sitting right on the structure they'd identified. Really helped nail down that these bumps were, in fact, the prohibitin complexes.
Okay, so we've gone from rings in a test tube to seeing these dome-like or maybe bell-shaped structures directly inside the mitochondria. What else did they figure out about the exact architecture?
They took it a step further with something called subtomogram averaging. You basically take lots and lots of these individual bump images from the cryo-ET data and average them together.
To get a clearer, more detailed picture.
Precisely. And this really confirmed that the structure in situ in its natural place is more like a bell shape.
A bell shape. Okay, that's different from a simple flat ring. What are the features of this bell?
Well, it's still about 20 nanometers wide at the base, the part interacting with the membrane.
Mm-hmm.
But then it tapers, narrowing towards the top that sticks out into the inner membrane space, down to about 8 or 9 nanometers across up there. And the whole thing is about 9 nanometers tall.
So wide base in the membrane, narrower opening facing the intermembrane space. Got it. How do the individual PHB1s and PHB2s fit together to make that shape?
That's where modeling comes in. Since we don't have a perfect experimental structure of the human proteins yet, they use predictions from AlphaFold, this AI tool that's amazing at predicting protein shapes.
Okay.
They basically tried to fit these predicted PHB1 and PHB2 shapes into the bell-shaped map they got from the cryo-EM averaging, like a molecular jigsaw puzzle.
And what did the puzzle look like when solved?
Their model suggests the bell is made of 11 prohibitin molecules total.
11, not 16 or 12, 20.
Seems like 11 fits the density best in this model. And they appear to be arranged alternating. PHB1, PHB2, PHB1, PHB2, and so on.
Okay, an 11 subunit alternating ring forming a bell?
Kinda, yeah.
Mm.
The model shows the transmembrane bits anchoring it in the membrane, the PHB domain sitting sort of perpendicular to the membrane, and those coiled coil regions we talked about.
The molecular Velcro.
Exactly. They seem to provide the main connections holding the 11 subunits together, stabilizing the whole bell structure.
Right. Those seem key. Did they have any other data to back up this 11 subunit Bell model?
They did. They looked at existing data from something called cross-linking mass spectrometry.
What does that do?
It helps map out which parts of proteins are physically close to each other inside a complex. They found that data from mouse and human prohibitins largely matched their model. The distances between linked parts made sense with the 11 subunit Bell shape.
Okay, so multiple lines of evidence, cryo-ET, averaging, modeling, cross-linking, all pointing towards this 11 subunit Bell structure.
Yeah, it's a much clearer picture than we had before.
All right, so now we have this detailed picture of the structure. Let's get to the really big question. What does this prohibitin complex actually do? Why is it there?
Ah, the function question. Well, it's involved in a surprising number of crucial mitochondrial jobs. One major role seems to be maintaining the structure of the cristae.
Keeping those folds folded correctly.
Exactly. Those folds are essential for maximizing the surface area for energy production for making ATP. If you lose prohibitins, the cristae morphology gets messed up. The folds can look abnormal. Think of it like internal scaffolding for those folds.
So critical for the power plant's physical structure. What else?
It goes deeper. They're also involved in maintaining the mitochondrial DNA, the mtDNA.
How so?
They seem to help organize and stabilize the mitochondrial nucleoids. That's how the mtDNA is packaged up with proteins inside the mitochondrion.
Okay.
So if you deplete prohibitins, you can run into problems with making new mitochondria and also with producing the proteins encoded by that mtDNA, many of which are vital parts of the energy production machinery itself.
Wow, so structure and maintaining the mitochondrial blueprints and machinery production. That's fundamental. Any other tasks?
Yep. They also seem to play roles in protein synthesis and degradation within the mitochondria, maybe acting as scaffolds or platforms.
Scaffolds for what?
For example, bringing together enzymes like MLA proteases. These are like the quality control crew, breaking down damaged or misfolded proteins.
Right, keeping things tidy.
Exactly. Now, the specific study we focused on didn't see them permanently stuck to MLA proteases in U2OS cells, but the interactions could be dynamic reporting. They might come together temporarily to do a job, then separate.
Ah, like a temporary workspace rather than a permanent fixture.
Precisely. And another big one. They seem necessary for properly assembling the IX-PHOS system.
The oxidative phosphorylation system, the main energy production line.
That's the one. They might interact directly with components like complex I or COX-2. If you reduce prohibitins, the activity of these energy-producing complexes can drop, and electron flow gets partially blocked. It gums up the works.
Okay, structure, DNA, protein quality control, energy production assembly, it's involved in a lot. What about the link to cell life and death? Apoptosis.
Yeah, that's another really fascinating area.
Right.
Prohibitins seem to influence whether a cell undergoes programmed cell death, apoptosis. If you have more prohibitin than usual, it can actually protect cells from apoptosis triggered via the mitochondria.
Protecting them.
By helping maintain the mitochondrial membrane potential, which is vital for energy, or by stopping the release of cytochrome C, a key death signal protein.
Right.
And maybe even by influencing survival genes. But flip side, lose prohibitin, and cells can become more sensitive to apoptotic signals. This might be linked to processing another protein, OPA1, involved in mitochondrial shape and fusion.
So it's like a rheostat dialing survival up or down depending on its levels or function?
That's a good way to put it. And it ties into stress responses too.
How so?
Well, under certain kinds of stress, like damage from reactive oxygen species, ROS, cells can actually increase their prohibitin levels. This suggests a protective role. And indeed, cells lacking enough prohibitin are more vulnerable to oxidative stress and cell death.
It really sounds like they're central players in mitochondrial quality control and overall health.
Absolutely. They touch on structure, genetics, protein handling, energy, cell fate. By influencing all these, they're key to keeping mitochondria healthy. And when they're not working right, the cell notices. There's a specific stress response called the UPRMT.
The mitochondrial unfolded protein response.
Exactly. That gets triggered when the prohibitin complex is lost or malfunctioning. It's like an alarm bell signaling that things aren't right in the powerhouse, further showing how important prohibitin is for preventing that stress in the first place.
Okay, that's an incredible range of functions for this one complex. Given all that, it makes sense that problems with prohibitin could lead to health issues.
That's precisely right. Because they're involved in so many basic mitochondrial processes, when they don't function correctly, it's been linked to a range of human diseases.
Like what, for example?
Yeah, things like neurodegenerative disorders, Alzheimer's, Parkinson's have been mentioned, and also various types of cancer.
Yeah, I can see how disrupting the power supply could hit energy-hungry brain cells hard, or how messed up mitochondria could play into cancer.
Exactly. And that's why getting these new structural details, like the bell shape, is so important. It gives us a foundation for understanding how prohibitin interacts with everything else and performs these jobs at a molecular level.
It's like having the blueprint for a critical machine part, finally.
Right. And now we can start asking how specific disease-linked mutations might break that machine.
So how does knowing it's an 11 subunit bell help figure out what goes wrong in disease?
Well, you can start to map where disease mutations occur on that structure. Does a mutation disrupt how the subunits fit together? Does it block interaction with, say, an OXB-HOS component or OPA1?
Yeah.
Understanding the precise defect.
Could lead to targeted therapies.
That's the hope. If you know exactly how the prohibitin complex is failing in a particular disease, maybe you could design a drug to, I don't know, stabilize the complex or restore a crucial interaction. It opens up possibilities.
That is really exciting, targeting these fundamental machines.
It is. It really shows how digging into the basic nuts and bolts of cell biology can eventually lead to potential new ways to treat complex diseases.
Okay, let's try and wrap this up for you, the learner. You've just taken a really deep dive into the prohibitin complex. It might seem like a tiny component, but it's clearly orchestrating a whole symphony inside your mitochondria.
Yeah, from its newly seen bell shape, which is pretty cool in itself, to being involved in energy, DNA maintenance, protein quality control, even deciding if a cell lives or dies.
Yeah.
It's way more than just simple scaffolding.
And what I find really fascinating is how something so fundamental, so deep in the cell's machinery, has such broad effects. It really shows how interconnected everything is at that tiny scale.
Absolutely. And understanding these intricate players, it's not just about satisfying curiosity, though that's part of it. It gives us vital clues for figuring out diseases that start with problems at this cellular level.
Think about it, these tiny bells covering maybe 1% of the surface area inside your cellular power plants are essential to keep them running. So the big question researchers are tackling now is, what happens when they don't run smoothly?
Right. And your knowledge now puts you right at the edge of understanding those ongoing discoveries. If you're really interested looking into the specific diseases linked to prohibitin problems, that's a fascinating rabbit hole to go down.
A deep dive for another day, perhaps. Thank you so much for joining us on this exploration of the biobits and bytes of the prohibitin complex.
Thanks for having me.
Until next time, keep that curiosity burning.